Having talked about the various principles and methodologies which are involved in obtaining a recombinant DNA and the technologies which are related to this entire process, now we are going to shift to a new topic, that is, what are the applications of this entire technology. Now, when we talk about applications, we first of all are dealing with our own self, that is application in human health, how our DNA technology is going to benefit the human beings. So, we have the application in human health, and our first topic is that of gene therapy. Now, what does this gene therapy means? You know, we as individuals have evolved from a long time, we are evolving, and we have seen many changes, and one certain thing that happens around us and inside us is that of mutation. Now, there are certain problems in human health that arise out of the changes at genetic level, which are certainly mutations are responsible for that. So, we have certain faulty genes inside us. Okay? That is why we see so many diseases in the human race. There are faulty genes which are responsible for certain diseases inside the human beings. So, this gene therapy aims at replacing this faulty gene with a healthy, normal functioning gene. And this process is known as gene therapy. Now, ranging from the simple single step mutations or a single nucleotide mutations, point mutations like sickle cell anemia, to the very severe combined immunodeficiency, that is SCID. Such diseases are being targeted under this gene therapy. Now, what we are going to consider is the example of SCID. It stands for severe combined immunodeficiency disorder. Now, the name is huge, hence abbreviated, but the root cause for such a long named disease is a faulty gene. Now, what is that faulty gene? That faulty gene is the gene which is responsible for ADA formation. Now, what is this ADA? It is adenosine deaminase. Ase is there, that means it must be an enzyme. This is an enzyme which is responsible for mounting the immune responses inside the body. Now, because of this, there is total metabolic pathway stoppage because it is not being formed inside the patients who suffer from SCID. This entire metabolic process which involves this adenosine deaminase is altered. Okay, it comes to a halt, it comes to a standstill, because of which the patient cannot mount an immune response. As a result of this deficiency, the T lymphocytes inside the patient are non-functional. Now, these T lymphocytes are very important class of immune cells which are responsible for telling the body what is self and what is non-self. Now, if the body is not able to recognize that the pathogen is self or non-self, how can it give any sort of immune response? And when the immune response is not there, you can very well imagine what would be the condition of the patient. He or she would be suffering a lot because of deficit immune system. Now, in that condition, what could be done? We can replace the faulty gene which is not working as it should be working under certain circumstances. That could be mutation, usually mutation, I would not give any other example. Because of mutation, the gene responsible for production of ADA has become the faulty gene. Now, what gene therapy was? We aimed at replacing this faulty gene with a functional gene, but this is not possible until and unless we try to replace that faulty gene inside the embryo itself in the stem cell stage. That is not possible. That is a very cumbersome process, and certainly it is it is very costly if practiced. Now, what is done as an alternative? What comes under gene therapy in this SCID is that we are going to use a vector whose own DNA would be compromised or subjugated, and we use the vector retrovirus. Retroviruses are used. These retroviruses have the recombinant DNA where we have the functional ADA gene incorporated inside them. Okay? The retrovirus is going to have the functional ADA gene inside it, but it is not going to have the genes which are responsible for its packaging. So, we are in a way cutting its pathogenic activity, and instead we are putting the functional ADA gene. Now, what is to be done? We extract the cells of bone marrow from the patient who is suffering from SCID. We take out those cells, or we take out the lymphocytes. Lymphocytes are taken out and cultured. The lymphocytes from the bone marrow of the patient are extracted. Okay, bone marrow cells happen to be stem cells. Lymphocytes would be extracted. They would be cultured with these retroviruses whose DNA has been altered by addition of functional ADA genes. Now, these retroviruses would go and infect the lymphocytes. When the lymphocytes would be infected, now because the virus has come, they would have the ability to produce ADA. Whatever was the deficiency, now the lymphocytes become properly functioning T lymphocytes, and once that has been achieved outside the body, inside the lab, in your culture tube or petri dish, you can put these lymphocytes back into the patient. Now, this is known as gene therapy. You took out the cells from the patient, you incorporated the non-functional gene, instead you put a functional gene at that place, and that gene is working inside the virus that happens to be the vector inside the cell, and you get gene therapy done. I am going to repeat it. That supposedly we have a patient suffering from SCID. Okay. Now, what do? From the bone marrow of SCID patient, we take lymphocytes which are non-functional. That was the problem. That is why the patient is having SCID, because the T lymphocytes are non-functional. They are not able to produce ADA. At the same time, what has been done, a retrovirus vector has been made a transformant. Okay, because it has got the ADA gene, the gene that is responsible for formation of ADA. Now, this ADA gene containing retrovirus would be made to infect the lymphocytes. Retroviruses have the tendency to infect, so it would be used to infect the lymphocytes. These lymphocytes would now become functional as they can produce ADA now. Where from they got the gene? They got the gene from the vector. Now, they produce ADA. Now, they can be put back into the patient. Okay, and now the patient can produce the lymphocytes. Now, this particular process is having certain complexities in itself, because you know lymphocytes have to be generated time and again. They are not immortal cells, they die after some time. So, the person has to be given this treatment after a gap of few years. At the same time, we do not only target SCID, there are many diseases that are to be targeted under gene therapy. So, till now this entire process of gene therapy is little bit tabooed and not completely explored because of the problems that are being faced because in this particular process. So, you have to keep in mind that this is one good application of recombinant DNA technology, and because it is one good application, you must know one good example for understanding the process, and once you have understood it, I would suggest you to learn it so that you get good marks, and whenever you appear for exam, you have an idea that what is being asked out of this application.